Hello everyone, myself Dr. Girisha, working as the assistant professor in Department of Plant Pathology, School of Agriculture, SR University. Today I'll discuss about integrated management of pomegranate diseases. Let's see about pomegranate crop. Pomegranate scientific name is Punica granatum, belongs to the family Punicaceae. Pomegranate is a native of Persia or Iran. Pomegranate is cultivated in home gardens, especially as a medicinal plant and as a fruit tree. There is no other fruit crop that has highest medicinal value compared to pomegranate, one of the oldest known fruit found in the writing aircrafts of many cultures and religions. This nutrient dense antioxidant rich fruit has been rewarded as a symbol of health, symbol of fertility, and symbol of eternal life. India ranks first in area with a production of 7 lakh ton but export only 30,000 ton. Maharashtra is the leading state in India, followed by Karnataka, Gujarat, and Andhra Pradesh, followed by Tamil Nadu, which are all major states producing the pomegranate. Ganesh, Bagwa, Ruby, Arcata, and Mridula are the important commercial varieties of pomegranate. This Ganesh and Bagwa are the major pomegranate varieties. Let's see about major diseases of pomegranate. One is bacterial disease, which is bacterial leaf and fruit spot caused by Xanthomonas axonopodis punicae. Then fungal disease, mainly leaf and fruit spot, anthracnose and wilt. This leaf and fruit spot caused by Pseudocercospora or Cercospora punicae. Anthracnose caused by Coltotrichum gliosporides. Wilt caused by Fusarium axisporum or Ceratocystis fimbriata. Coming to post-harvest disease, mainly grey mold, Aspergillus decay, and alternary rot. So these are all the important pathogen affecting the pomegranate crop. Let's see about one of the important diseases in pomegranate. It's the bacterial disease, bacterial blight of pomegranate. So this bacterial blight, which is caused by Xanthomonas axonopodis pethoverpunicae. Coming to this etiology, this is caused by the gram-negative bacteria, rod-shaped bacteria, aerobic and single folar flagellum, yellow-colored mucoid colonies, and it will always get entry through wounds and natural opening because these bacteria are passive parasite. They don't have any own structure to penetrate the plant cell wall, so they always enter through stomata or wounds or any other natural opening like stomata or any other natural openings present in the plant because of their passive mode of entry. So coming to this important, it's economic important. It is found in the state of Maharashtra, Karnataka, Andhra Pradesh. In Karnataka, major districts affecting are Bagalkot, Gadag, Koppal, Ballari, Chitradurga, and Bijapur area. Around 60 to 80% loss is reported, even 90% reported in some areas of Karnataka. Even 100% severity is reported in some of the orchards in Maharashtra. So it is one of the important severe disease affecting the pomegranate growers nowadays. Coming to the major symptoms of bacterial blight of pomegranate, blight affect all the parts and such as symptoms are observed in leaves, stem, flower and fruits. Coming to the symptoms on leaves, minute spherical water-soaked lesions are observed on the foliage, which later become a dark brown black with a necrotic center surrounded by translucent halo. In advanced lesion, however, the translucent halo may not be visible. A lesion may be coalesced and often extended to veins and midrib also. Infected foliage normally turns yellow and fall off prematurely. The premature defoliation is the major symptom of bacterial blight on the leaves. So these are the pictures representing the water-soaked lesion with the necrotic spots and surrounded by yellow zona, yellow halo. Coming to the symptom of stem, the twigs and stems reveal a brownish black lesion, generally initiating at the nodes and extending along the bark of the stem. Infections are normally observed in the bark cortex region and also extend into the vascular region of the plant. Blight lesion on the twigs and often result in girdling. Stem girdling is a major symptom, thereby resulting in breaking of the nodes twig at the point of infection. Such twig normally reveal the drying with yellowing of the leaves and remain attached to the plant until they become detached by some external pressure like wind or water. Since the blight infections are prominent at the nodes and the disease is known as nodal blight. So this is the picture representing the nodal blight symptom. You can see the fruits remain attached to the infected plant and complete defoliation occurs, and severe nodal blight symptom we can observe in the infected plant. Coming to the infection on the fruit, so this is a major symptom. Initially, blight symptom on the fruit appear as a small water-soaked lesion which increase in the size and turn brownish black and necrotic. Lesions in the fruit often reveal X or Y or L-shaped small fissure or small splitting on the infected fruit, which are generally not observed in spots caused by some fungal pathogen. So this is one of the unique character caused by the bacteria. Legends in the fruits normally coalesce and many results in the blighting symptom. The blight fruits which with one or more legends reveal a characteristic splitting and rendering the fruits which are unfit for consumption, which are unfit for marketing. In the picture, we can see the young fruit infected with the water-soaked lesion with the necrotic spots. And in another picture, we can see these two pictures. There is a characteristic splitting in the Y shape or L shaped fissure formation on the infected mature fruit. So this is the major symptom which is affecting the economic part of the pomegranate. So it will completely reduce the market value and unfit for consumption. Coming to survival and spread of the bacteria, the primary source of inoculum is through infected cuttings. Here, the bacteria they don't have any spores or unlike the fungi. The major mode of entry is through passive mode of entry through natural openings like stomato. Then secondary source it will spread through wind splash and rain splash, pruning tools, humans and other insect vectors. Coming to favorable condition, increased day temperature of 38 degree and afternoon relative humidity of 30%, along with a cloudy weather and intermittent rainfall, favor the disease initiation and further spread of the disease. Coming to bacterial blight pomegranate disease cycle, mainly the primary infection occurs by the bacterial cell present in the infected cuttings. Then they will cause the symptom like fruit splitting, then the symptom on the leaves and the foliage part, then the secondary spreading of this bacterial cell mainly by rain splashes or wind or any pruning tools or humans and other insect vectors. Then the bacteria survive in the soil for as a saprophyte in the plant debris on the fallen leaves and infected fruit parts. Again it will cause infection to the young new plants. So this is how it will complete the life cycle. So coming to management of bacterial blight of pomegranate, one of the important disease, let's see about cultural practice, wide row spacing, selection of the disease-free seedlings for the fresh planting. We can go for tissue culture planting, pruning affected branches, fruits regularly, and burning of the infected plant debris. Bahar should be done in Asta or Ambe Bahar. Give minimum 4 month rest after harvesting the fruit. Coming to biological control, apply biocontrol agent like Bacillus subtilis, Pseudomonas fluorescens, and Trichoderma archanum. Soak neem leaves in a cow urine and spray in order to control the pest and plant pathogens. Apply 40% tulsi leaf extract followed by neem seed oil. Also apply an extract of garlic bulb, meswak stem extract, and patchouli leaf extract at 30% concentration. Coming to chemical methods, before pruning, it should be sprayed with a 1% Borax mixture. You know how to prepare the Borax mixture? Like 1% Borax mixture should be in the ratio of 1 is to 1 is to 100, 1 kg of lime, 1 kg of copper sulfate dissolved in 100 liter of water. The pH should be neutral. After ethyl spraying or defoliation, paste or smear with the 0.5 gram streptomycin sulfate plus 2.5 gram copper oxychloride and 200 gram of redoxide per liter of water. Spray 0.5 gram streptomycin sulfate or bacterionacetic or 2.5 gram copper oxychloride per liter of water. Next day or another day, spray with 1 gram zinc sulfate plus 1 gram magnesium sulfate plus 1 gram boron plus 1 gram of calcium sulfate per liter of water. Pruning tool should be disinfected with a suitable disinfectant like sodium hypochlorite 1.5 to 2% before pruning the new plant. Use resistant variety play important role in the management of any disease. Here, like our all the present popular varieties like Ganesh, Bhagwar, Gata, Mridula, Ruby, which are all susceptible to bacterial blight. Breeding work on the pomegranate has been in progress at various NRCs and research centers in the country to develop suitable blight-resistant varieties. So this is about bacterial blight of pomegranate caused by Xanthomonas axonopodis, Pethovorpunica and its integrated management. And let's coming to another important disease which is anthracnose caused by Coltotrichum gliosporides. Coming to the major symptom of anthracnose in pomegranate appears as a small regular or irregular dull violet or black leaf spot which are yellowish halo. Leaves turn to yellow and fall out. Symptoms appear in the flower also. Both render and mature fruits develop a spot which are initially circular, later become irregular, brown to dark brown in color, covering the entire fruit, only with a sunken center. Disease portion appear with a minute black dots, which are representing the acer uli. So these acer uli are nothing but a asexual fruiting structures of the fungus, Coltotrichum gliosporides. It contains conidiospores and conidia that will spread the disease. So these are the pictures representing the formation of small irregular black leaf spot on the infected foliage and dark black color necrotic spots, the sunken center present on the infected fruits. Coming to favorable condition for anthracnose of pomegranate, the disease is severe during August, September when there is a high humidity and temperature between 20 to 27. Coming to survival and spread of the fungus, primary source of inoculum is infected leaves. The secondary spreading of inoculum with infected wind-borne conidia. It also infects the mango, guava and papaya. So this is the disease cycle of anthracnose of pomegranate. The primary infections occurs by the spore present in the soil and dormant mycelium present in the infected plant debris. Then formation of the symptom on the foliage and infected fruit. And secondary infection through conidia, which are present on acerulae, asexual fruiting body of the asexual fruiting structure of the fungus Coltotrichum. Then it will carry through wind and rain splash, and again the pathogen will survive on the infected plant debris or in the infected soil. Again it will cause the primary infection. Like this how it will complete the disease cycle in of anthracnose of pomegranate. Coming to integrated management of anthracnose of pomegranate, first is cultural method, field sanitization, destruction of the infected fruits, selection of resistant varieties. Coming to mechanical method, proper sanitation of farm implements. Then coming to major like chemical methods, carbendazim, difenoconazole or thiophanate methyl at the rate of 0.25 ml per liter, which can effectively control the disease at fortnightly intervals. Have found to be effective. Spraying of difenoconazole 25 EC at the rate of 1 ml per liter and like other fungicides like chlorothalonil or carbendazim also found to be effective against the anthracnose disease. Spraying some systemic insecticide like hexaconazole 1 ml per liter, thiophanate methyl 1 gram per liter or carbendazim 1 gram per liter at 20 days interval, it is quite effective. Among the contact fungicide, chlorothalonil and mancozeb at the rate of 2 gram per liter were found to be more efficacious. So this is regarding the cultural, mechanical and chemical management of anthracnose of pomegranate caused by Cultotrichum gliosporides. Let's coming to another important disease, leaf and fruit spot of pomegranate caused by Cercospora punicae. First coming to the symptom of leaf and fruit spot of pomegranate. Leaf spots are minute, brown with yellow halo. Spots are scattered, circular or irregular and become dark brown with the aging of the plant. Spots on lower side are sunken with a cluster of spore bearing structure and is grayish in color. Minute circular black spots also appears on the sepal of the flower. It will also infect the flower. Fruit spots are black, minute, circular, and which are majorly concentrated on the rind region of the fruit. When grow old, they become large, irregularly circular and depressed, presenting an ugly look to the fruit which is unfit for consumption and which will greatly reduce the market value of the fruit. So these are the picture representing the formation of minute brown to black color spots with a yellow halo present on the infected leaf, and also the formation of the spots on the infected flower and fruit also. Coming to favorable condition, optimum temperature is 25 to 32 with highest temperature above 16 degree and relative humidity of 90 to 95%. The disease is serious during some September to November, and survival and spread. Primary spread is through disease plant debris and secondary is through wind-borne Cundia. Uh, this is the disease cycle representing the leaf and fruit part of pomegranate caused by Cercospora species. So here mainly the primary infection occurs by the spores present in the soil or infected plant debris. Mainly these Cercospora serves as a saprophyte in the infected plant debris. Then expression of the symptom on the fruit and flower. Then secondary infection by conidia through rain or wind. So this is the conodial structure of Cercospora, which will produce the slender hyaline, which is having 7 to very thin hyaline 7 to 8 celled conidia. Then again the secondary spreading is through wind or rain, and then later on it will survive on the infected plant debris or infected soil. Coming to management of this disease, coming to cultural management, the disease fruit should be collected and destroyed. Pruning and destruction of the disease twigs, spray the crop with hexaconazole 5 EC or propiconazole 25 EC or difenoconazole 25 EC at the rate of 1 ml per liter. All these are systemic triazole fungicides. Or quitazine 48 EC at 2 ml per liter, or carbendazim 1 gram per liter, or chlorothalonil 2.5 gram per liter. Or you can also go for copper oxychloride 50 WP at the rate of 3 gram per liter. Thiophanate metal and carbendazim at the rate of 1 gram per liter is most effective. Among the contact fungicide, chlorothalonil 2 ml per liter followed by mancozeb 2 gram were most efficacious. Coming to wilt of pomegranate is the one of the major disease affecting the pomegranate cultivation, which is caused by Ceratocystis fimbriata and Fusarium oxysporum, which is a complex disease. There is involvement of nematode, root knot nematode also. We'll see one by one. First is the symptom. Affected plant shows yellowing of leaves in some twigs or branches, followed by drooping and drying of the leaves. So this is the above ground part symptom. The entire tree dies in a few months or in a year due to infection by wilt causing pathogen. When the affected tree is cut open in the lengthwise or in the longitudinal cross section, we can see dark grayish or brown color discoloration of the woody stem is seen mainly due to infection to vascular tissues like xylem and phloem tissue. Wilt infected plant often reveal a dried foliage and fruit attached to the plant branches for many months. So this is the image representing the wilt affected plant, yellowing, drying and drooping of the leaves. When we split open the infected plant parts, when we cut the plant in a longitudinal section, we can see brown color discoloration with a complete drying and wilting symptom on the infected plant. Coming to other causes of wilt, one is already know Ceratocystis fimbriata, followed by Fusarium oxysporum or Fusarium solani. A dry root rot disease of pomegranate reported from Maharashtra and India was found to be Fusarium solani. Reported pomegranate wilt due to Fusarium oxysporum from the Ahmednagar and Solapur district of Maharashtra. Macrophomina fasciolina, which is another pathogen involved in causing the wilt disease in pomegranate. The pathogen has isolated from the root and collar portion of the wilt affected plant, causing the rotting of the roots, damping of, thereby resulting in wilting of plants. Coming to Meldogina, which is a root knot nematode, Verma in 1985 reported the susceptibility of some pomegranate varieties to Meldogina incognita. Studies of some scientists revealed that 33% yield loss it is mainly due to root knot nematode. In these nematodes are, we can call it as hidden enemies. In some orchards, nursery plant, critical examination of the roots of wilt affected plant revealed a development of knots on the main and secondary root, which in turn revealed the association of eggs of and adults of Meldogina incognita, as the report by Sharma in 2008. Coming to survival and spread, the wilt pathogen, Ceratocystis fimbriata, probably survives adverse condition as mycelia within the plant host or as a thick-walled Aliroconidia, which are present in the soil, or infected plant host or infected plant debris. The fungus survives in the pomegranate plant pots up to 190 days and in soil for at least 4 months. Infection of the above ground plant organs by such wilt pathogen usually takes place through wounds. However, the infections have been observed in the soft tissues under artificial condition. Some studies carried by NRCP Solapur revealed that Ceratocystis fimbriata could be infect the roots of plant directly without any presence of wound also. Primary source of inoculum is soil and secondary inoculum are conidia and water. Somashekar and Wali indicated the role of scalded beetle in the spread of the pathogen Ceratocystis fimbriata. The disease is more severe in heavy soil and increase with the soil moisture. This is the wilt life cycle. This is the life cycle of Fusarium oxysporum. Fusarium oxysporum will produce microconidia, and chlamydospore. The primary infection is caused by the spores present in the soil, and the secondary infection or secondary spreading is caused by micro and macroconidia through irrigation water or rainwater. Again, the fungus will produce chlamydospore, very thick walled or resting spores, which can survive in the soil for longer duration. Coming to management of wilt of pomegranate, first is cultural method. Don't allow water to stagnate and try to create drainage facility. Do not irrigate for 2 to 3 days after drenching, and soil application of bacillus cultures like Bacillus subtilis was effective in the field condition, reducing the pomegranate wilt indicate incidence due to Cercospora fimbriata. Soil application of Trichoderma plus Bacillomyces species at 25 gram with a 2 kg well decomposed FIM format manure around the trunk of pomegranate tree helps to prevent the wilt infection. Application of Bacillomyces laciniens and Pseudomonas fluorescens at 10 to 20 gram per meter square resulted in the effective reduction of the root knot nematode population. Trichoderma everyday bioformulation resulted in 86.6% growth inhibition of Ceratocystis fimbriata under in vitro condition. Coming to chemical management, at the initial stage, drench the plant with propiconazole plus 4 ml of chloropyrifos per liter of water. Drench 8 to 10 liter of solution per tree. If any plant already wilted, drench chemical as per the above mentioned, then remove the entire plant along with the root and burn it out. Drench with the formaldehyde at the rate of 25 ml per liter is also found to be effective. So this is regarding the wilt disease, complex disease of pomegranate caused by Ceratocystis fimbriata, Fusarium, and association with the root knot nematode. Coming to other disease like fruit rot of pomegranate, fruit spot, which is caused by Alternaria alternata. Coming to the symptom, small reddish brown circular spots appears on the fruit. As the disease advances, these spots coalesce to form larger patches and fruit start rotting. The aerials get affected with become pale and become unfit for consumption. Coming to management, cultural method like field sanitation, growing resistant varieties, and chemical methods like application of mancozeb are capped off at the rate of 0.25%. Coming to scab disease of pomegranate, which is caused by Specialema punicae. Coming to the symptom, infected fruit become distorted and may crack, allowing the entry of secondary parasites. Scab can also infect the flower stem and cause flower to drops. Coming to management, first is cultural method. Choose resistant variety, destroy infected leaves to reduce the number of fungal spore. Water in the evening early hours to avoid the overhead irrigation. Spray 3 to 6 inch layer of spreading this 3 to 6 inch layer of compost under the tree, keeping it away from the trunk to cover the soil to prevent splash dispersal of the fungal spore. And chemical method we can apply for go for the difenoconazole at the rate of 0.5%, which is the triazole fungicide. It has more efficacy to control the scab caused by Specialema punicae. So that's regarding some important fungal and bacterial disease in pomegranate caused by different fungal pathogen. These are some IDM practice recommended for important diseases in pomegranate, starting from the nursery area till harvesting of the fruit. In nursery raising and preparation of the planting material, approved or certified nursery must be obtained the standard planting material, air layer, hardwood cuttings or tissue culture sapling from the respective progeny orchards. To prepare approximately 100 kg of potting material, mix 50 kg of soil and 50 kg of compost along with a consortium having 1 kg each of Trichoderma arceanum, Azospirillum and 5 kg of neem cape. Planting should be done after 8 days of mixing the above ingredient. Apply Borax paste 10% to cut ends of the mother plant and air layered cuttings. Treat the roots of hairs with air laid cuttings with the copper oxychloride at the rate of 3 gram per liter to protect against the soil-borne diseases at the seedling stage, and plant them in a standard size polythene bag filled with the above potting mixture. Place planting material under 50% shade net. Monthly spray application of copper oxychloride along with the streptocycline altered with the bronopal, which is highly recommended to control bacterial blight affected area. If the fungal leaf spot appear, use the combi product fungicide mancozeb plus carbendazim 2 gram per liter or chlorothalonil 75 WP 2 gram per liter. We can repeat the same application at the rate of 15 days interval. Planting and care of new orchard. Select the site having the light soil for establishing the orchard. Make the pits 1 meter deep at the row to row spacing of 4.5 meter and plant to plant 3 meter. Pits should be dug at least 1 month prior to planting and keep it open, disinfectant the pits with intense solar radiation during the day. Just before filling the pit, drench the bottom sides of the pit with 4 to 5 liter of 0.4% of chloropyrifos 20 solution. Dust the pit with the bleaching powder at the rate of 33% chlorine at 100 gram per pit before filling the pit. In each pit, mix the following topsoil with the FIM or vermicompost or neem cake and bioinoculants. Before taking and planting material to the minefield, spray the plant with the copper oxychloride 2.5 gram per liter plus streptocycline 0.25 gram per liter. Care of pre-bearing orchard up to first year of planting. 1 month after planting, spray with the streptocycline 0.5 gram per liter plus copper oxychloride 2.5 gram per liter in epidemic areas only. COC in disease-free areas is recommended. Take alternate spray with the Borax mixture 1%. Alternatively, COC and streptocycline spray have to be replaced with the bronopal at the rate of 0.5 gram per liter plus capped on 3 gram per liter. Spray interval should be 1 month in disease-free orchard. In orchard where the blight incidence is observed, spray schedule should be 15 days during dry periods and 7 to 10 days during rainy days. In orchards having the blight, one spray with the streptocycline 0.5 gram per liter plus copper oxychloride 2.5 gram per liter after rain stops is mandatory. If the fungal leaf spot appear as a spray with a combi product fungicide, carbendazim plus mancozeb or chlorothalonil or hexaconazole, we can repeat the same application at the 15 days interval. Monitor regularly for blight symptom. In case of observation of the foliar symptom, recommend spray out to be commenced if the blight symptoms are observed on the stem. Prune and remove the infected twigs as and when the symptom is noticed. Any severely infected plant has to be removed and burnt and replaced with a new disease-free plant. Coming to establishment of orchard after second year, in the bacterial blight prune areas only Asta Bahar or late Asta Bahar crop must be regulated. In bacterial blight area, grower may take Bahar conveyed to them. Coming to the rest period, as a rest period of 4 months is recommended for better health vigor and better fruit bearing capacity and also reduction of the pathogen load. As the rest period falls during rainy season, spray 15 days interval of Borax mixture 1% altered with a spray of streptocycline 0.5 gram per liter plus copper oxychloride 2.5 gram per liter or bronopal at the rate of 0.5 gram per liter plus captan 3 gram per liter in epidemic areas. No manures and inorganic fertilizer is recommended. Minimum irrigation is preferable. Remove the water shoots regularly. In orchards where the severe blight infection is noticed, go for heavy pruning immediately after harvest and remove as far as possible all the stem with a fresh blight infection. Prune out 2 inch below the infected area. Canker, if any, should be scooped out till normal wood appears on them and paste with Borax paste 10% at the cut end after pruning and scooped canker areas. Oil-based paste or COC made by mixing 500 gram COC, 1 liter linseed oil or chabutia paste prepared by mixing 1 kg of red lead or 1 kg of plus 1 kg of copper carbonate in 1.2 liter of linseed oil is preferred for pasting during rainy season. Any severely infected plant must be uprooted, burnt, or replaced with a new disease-free plant or cut from the base 2 to 3 inches below the ground level. New well-grown sprouts have to be trained for a new disease-free plant. In disease-free orchards, practice need to be based on pruning after the rest period. Spray Borax mixture 1% immediately after pruning. At the time of harvest, pomegranate are easy to harvest, require minimal ladder work. Fruits are harvested by clipping them with the shears. Cut as close as to the fruit possible to prevent the sharp point of woody from piercing and rubbing against other fruit in the bin. Either where the fruit should be handled with care in order to minimize the scupping or cracking when we are transporting the fruits. Managing the field condition, no pre-harvest fungicide treatment are available effective in preventing all the disease. The clean culture, removal of dead branches, mummified fruit from the orchard, trying the pickers or graders to exclude internal decay of fruit during harvest and packing. Shaking of branches by hand prior to harvest may remove potentially diseased fruit. Post-harvest storage, fruit destined for fresh market should be washed with chlorine, rinsed with water, sorted by culls, cracks, defects, or color and size and weight of the fruit. A storage bag we can also apply to promote the visual quality of the fruit and increase the storage life by reducing the moisture loss. In storage, fruit can be stored at 5 degrees Celsius for 90 to 95%. Relative humidity of 2 months. In case of storage beyond 2 months, temperature should be maintained at 10 degrees Celsius to avoid chilling injury. Pomegranates are very susceptible to water loss, resulting in shriveling of skins. Storing fruit in a plastic liner with a vaccine can reduce the water loss, especially under conditions of low relative humidity. Coming to fruit sanitation, coming to physical method, pomegranate is dipped into hot water 45 degrees Celsius for 4 minutes. Chilling injury symptoms are significantly reduced. Coming to chemical, after bin dumping, fruit are being washed with a chlorine. No phytotoxicity on fruit at the rate of 200 ppm chlorine solution. Fruit dip in fluoxetine 5 mg per liter will be control the gray mold disease in pomegranate. So here I have given one PDF given by National Research Center, uh, developed by National Research Center, Pomegranate, Solapur, Maharashtra. Here we can see integrated disease and insect pest management schedule for pomegranate cultivation, which is released in 2023. Here the given different schedules and different chemicals for the effective management of different pest and disease in pomegranate. And also here I have given some important objective questions related to important disease in pomegranate, like wilt disease of pomegranate is caused by fungus. The primary pathogen responsible for the wilt pomegranate is Fusarium oxysporum. The common symptom of wilt is sudden drying and wilting of the branches. Which practice helps to manage the wilt disease in pomegranate? Answer is use of disease-free planting material. Soil drenching with the fungicide is recommended for wilt disease. Bacterial blight of the pomegranate is caused by Xanthomonas oxynopodis, Pethover. Bacterial blight mainly affects leaf, stem, and fruit. Mainly, it will affect the fruits and development of cracks on the fruit. A typical symptom of bacterial blight is water-soaked lesion turning dark brown color. Which is effective management strategy for bacterial blight? Antibiotic plus copper-based application. Pruning and destruction of the infected plant parts helps to control bacterial blight. Leaf spot in pomegranate is commonly caused by Alternaria alternata. Symptoms of the leaf spot include like small brown or black spots on the leaves. Which condition favor the leaf spot disease? High humidity will always favor the leaf spot. Management of leaf spot include use of fungicide. Sanitation practice help to reduce the spreading of the disease. Which chemical is commonly recommended to control the fruit spot, which is nothing but carbendazim. Copper oxychloride mainly used to control bacterial disease in pomegranate. Water logging should be avoided to manage wilt disease in pomegranate. Integrated disease management include a combination of culture, biological and chemical methods. Resistant varieties helps in reducing the disease severity in pomegranate. And also we have given some theory based questions related to important disease in pomegranate. Thank you.